So my article, the title was Scaffold-Mediated Lentiviral Transduction for Functional Tissue Engineering of Cartilage, or in simpler terms, it's rebuilding cartilage. And just to acknowledge the people who helped me, Mr. Dean, Sydney, thank you for calling Duke University to get this article, and Mark and Jackson and Shelley, thanks for helping me with the presentation. But before I get to the goal, I just want to give you a little background. As some of you may or may not have known, I tore my ACL. I actually tore it twice, as you probably have seen me walking around the school with this big-ass brace on my leg. And this study pretty much study why ACLs fail or why cartilage rips. And for ACLs, there is a 10% failure rate, so 90% of the time, your ACL will successfully be replaced and you won't have any problems with it. Except for me, I was the special 10%, where my ACL actually failed the second time and they had to replace it with something else. So this study studies why that failed. And they also think of new ways or new methods to make repair better. And so the goal of the study was pretty much to successfully repair ligaments or tendon injuries, except in this specific paper they just focused on cartilage, but this could lead to tendons and ligaments. And I, for example, did the ACL. I'm focusing on the ACL because that's something close to me. And they wanted to develop tissue and promote rapid tissue repair. So cartilage is the flexible soft tissue found in between your bones, and they believe that if they could regrow this cartilage, and because cartilage can't grow itself, then maybe they can pass this on to ACLs or MCLs, PCLs, anything else. So pretty much successfully grow cartilage. So how did they do it? They did it through something called gene therapy. There's 3 methods for gene therapy, and it's used as a therapeutic way to replace DNA or to inject DNA into your body. And so the 3 different ways are knocking out a mutated gene, replacing a mutated gene in your cell, for example, cancer or any other diseases. But for this specifically, they introduced a whole new gene into your cell. And they did this through something called gene therapy, which is ex vivo, which means they physically took the cells out of your body and put them on little tiny dishes, and then they grew the cartilage on mediated scaffolds, and which you would put back into your body. But just for this experiment, they didn't put it back into human bodies. They just wanted to see if it would work. Okay, so where they started. They used hollow lentiviruses to deliver TGF-beta3. TGF-beta3 is a protein that promotes cartilage growth. And lentiviruses, it's physically a virus, and what they do is lentiviruses can introduce a large amount of DNA into your body. So they put this specific protein, TGF-beta3, which promotes cartilage growth, into lentiviruses, and they wanted to inject them into the cells. But they wanted to make sure that lentiviruses were the best possible method for this. So lentiviruses, what they did was they had 2 experiments where they had cells where they just pre-transduced TGF-beta3, which means they just introduced this growth factor. Lentiviruses was when they physically injected a virus into the cells. Now this is important because viruses, it is a virus. So they wanted to see if this would actually produce more TGF-beta3 than would just introducing the protein into the cell. And as you can see, lentiviruses is it looks like a virus. And from this graph, the black shows the lentiviruses, and it shows that there was more of an increase with when cells were injected with lentiviruses than if they were just introduced it, pre-transduced. Does everybody get that? Yes.
TGF-beta3 do again?
TGF-beta3 is the growth factor for cartilage growth. So pretty much that's what grows cartilage. Any other questions? Okay. So pretty much the more TGF-beta3 you have, the more cartilage you have. So for this example, they wanted to see for the 2 different things that they focused on was NT, which means non-transduced. This is your natural cells. Nothing is introduced to it. And then they also had cells that had lentiviruses introduced to it, which had the TGF-beta3 in it. And this study proved, this experiment, that that cells that did not have extra TGF-beta3 introduced in it, they didn't grow any more cartilage. But as you can see with the huge increase that when they introduced the cells with these lentiviruses, there was a huge increase in TGF-beta3, which is about 1,000 times increase, which promoted cartilage growth because cartilage can't grow by itself. So this proves that with the lentiviruses, it does promote cartilage growth. And they did this ex vivo on Petri dishes. Does anybody have any questions about that? No? Okay. So why choose lentiviruses again? So the advantages of the lentiviruses is it doesn't just promote cartilage growth, but it pretty much grows everything else that comes with the cartilage. So ACAN and COL2A1 are proteins that are also important for this cartilage growth. So when you grow cartilage, you kind of want to grow everything together. They didn't just want to single one thing out, but they did see, again, just focus on the red, which is the non-transduced, and then the cells that were introduced with the lentiviruses. It shows that when the lentiviruses were introduced, the other proteins that were important for cartilage growth also grew with it, which made it stronger. And then here, it's the GAG, which is glycosaminoglycan. All you really need to know is it's a huge sugar, and this acts as the lubricant or shock absorber between cartilages. So, for example, if you're pivoting or if you stop, jolt right, like after running or anything, this pretty much absorbs the shock. And if you don't have this sugar, this is very important because then the cartilage can rip or it's more open to injury. So this is very important because, as you can see, there was a huge increase even as time went on, even within 2 weeks, that this sugar grew with the cartilage. And this is very important. Any questions? Yeah?
What's the RHT?
The RHT, that was just, they pretty much just, it's not that important for the study. They pretty much just introduced the TGF-beta3, but they just produced a lot of it. It wasn't really even important in this. All you really need to focus on was the NT and the lentiviral. Because this pretty much has the same as lentiviruses. It's not really that important.
So your point with this slide is just that you're producing accessory proteins with the cartilage.
Yes. Which you pretty much want everything to grow together so that the cartilage is more strong. Yeah, what's up?
So looking at the second graph, the white bar is ACAN.
Oh, I'm sorry. Good question. Okay, so this is ACAN. This is ACAN, this is COL2A. These are two important proteins. For this is just focusing on the SGAG, which is the big sugar. And this is the non-transduced and the LDT. They just wanted to measure the amount in each of the cells. Any other questions? Okay. So pretty much this isn't your natural cartilage. They're physically putting something else in your body. This is mimicking. That's why ACLs or tendons or ligaments, they fail because it isn't what you were born with. So they wanted to make sure that this cartilage was appropriate, specifically cartilage, was appropriate and strong enough. And so what they did with this graph pretty much shows the aggregate modulus, which is pretty much the measure of stiffness of that cartilage. They wanted to make sure that it was, even though we're producing more, they want to make sure that it was just as strong as your native cartilage that you were born with. So in these graphs, I need to know in this one, the dotted line means day 0, that was from the beginning. And there's no significant difference between the cartilage you were born with, which is the NT, so non-transduced, these are your natural cells, and the cartilage that they created. Does everybody understand that? Because this proves that the cartilage that they create, this mimic cartilage, isn't weaker. It's actually just as strong. Yes? Okay. And then the hydraulic permeability describes the ease with which fluid passes through the cartilage. And this is also important because you don't want a very permeable cartilage. Otherwise, it could be, again, open to injury, or you want it to be kind of thick. And so with this one, on the side, it's actually approaching 0, so it's kind of actually going up in the negative. So it shows even here, So this was the non-transduced cells, and then this was the cells introduced with the lentiviruses. And this is actually very cool because it proves that even this new cartilage that they created is actually less permeable than your original. So this is actually stronger because they're producing more and they're getting more out of this mimicking cartilage. This cartilage can actually be stronger than the cartilage you're born with. Everybody get that? Any questions? Okay, because this is actually like the end of my presentation. Okay, and then, so why is this important? Yeah, that went by fast. Okay, so why is this important? So pretty much this opens a whole new study for a promising strategy for cartilage growth. Because as you know, tendon tears, like when you're older and your cartilage is getting worn out, or even like if you're an athlete, because I tore my ACL when I was 14 years old, this is very cool because this is the beginning of a study where maybe they can use gene therapy to inject your tendons or ligaments, which can get you a better recovery process and hopefully decrease the chances of re-injury. And so this pretty much opens a whole new avenue, and this is not perfected because this study specifically just focused on cartilage, but this interests me because this could maybe hopefully decrease that 10%, that was me, unfortunately, to hopefully get a better outcome with ACL, MCL, or any other ligament repairs. Yeah, sorry, I think
Did they mention any future plans to go and study other types of?
they didn't focus that on the study. Any other questions? Yeah.
Holly, this is just a personal question. After your first ACL repair, was the therapy and the strengthening different when you had the second one?
Yes, because the first time I used my patella tendon, which came right from my kneecap, and well, it's like right below my kneecap, the patella, and that takes longer to heal. So about ACL, it takes 7 to 12 months to fully get, well, it heals in about 6 weeks, but for you to fully get back up, it takes about 7 to 12 months. And the first time it was a little bit of a harder rehab, but this time they used my hamstring, which, they used my hamstring, and it has a lesser success rate. So last time with my patella tendon, I had a 10%, so it was a 90% success rate, and now this one that I have right now is an 80%. And so the rehab was a little bit different. The hamstring was actually easier, but supposedly the patella is supposed to be stronger, but I don't do that. Go ahead.
Did you ever consider a cadaver and
Yes, we did. However, because I was only 14 at the time, putting a dead person's body, ACL into my body was a little bit much. So they said, we're going to use what you have. There's also different ways. You can also use your quad, but it's a little bigger. Usually people don't come back from that recovery. And you can ask me personal questions about it. Go ahead.
Did they talk about why they use that specific kind of gene therapy?
Because lentivirus because it can introduce a huge amount of GNA into the cell, that seemed like the best possible method for introducing that type of gene therapy. Why they use gene therapy? Because they wanted to see if they could almost introduce your genes or more growth factor genes to hopefully grow the cartilage. Because tendons, tendons, ligaments, cartilage, they can't grow by themselves. It's one of the only things in your body where blood does not run through. You have to repair. So they wanted to almost promote growth and shove more genes into it to hopefully make it grow.
And they couldn't just overexpress a pre-existing?
I'm not sure about that, but I know that for this study, that's just what they did. Is your question? Yeah.
Does the gene therapy use the same transcription factors that your body would normally use to produce cartilage?
Cartilage, the problem is cartilage can't regrow itself. So as you can see, I kind of clicked it quicker, but there's not, when you just have a non-transduced, there's no increase in growth. All the grafts with the non-transduced cells are very small because cartilage can't grow itself. So for here, it's all very small because barely any TGF-beta3 was being produced because your body can't naturally.
So why exactly is the cartilage from the gene therapy stronger?
It's not necessarily stronger, but it produces a larger amount.
Oh.
Okay, yeah.
Is there a reason they only focused on cartilage? Did they think growing tendons would be more difficult?
They started with cartilage. I'm not 100% sure why, but this was kind of the first step. But they did mention in the article that this study, because if they can master down growing cartilage, it can lead to tendons and ligaments because they're very related. They didn't mention any side effects because they're only injecting, because it was a virus and that's what they were scared of, but they were only injecting a growth factor. So there wasn't, and also they only did this on Petri dishes and then on mediated scaffolds, which you would then put into your body. But they just wanted to make sure that, okay, it's growing cartilage and it's healthy and it's strong.
Would this externally grown cartilage take in your body just as well as stuff taken from other parts of your body?
I'm not 100% sure. They always say that using things from your body is almost better because it's your body, but this hopefully even people in their 70s or 80s who have a lack of cartilage growth, this could really help them.
So does your body naturally have the whatever the protein is, and then this was just putting more?
Yes, it does have the growth factor because you're born with cartilage and you can grow a little bit of cartilage, but when you tear it or it gets destroyed and somehow it can't fully repair itself. Any other questions?
So this isn't gene therapy in the typical sense, where they would, attempt to infect a human with this.
Correct.
They're just trying to build cartilage forms.
Right, because the other two types of gene therapies are replacing a mutated gene. So if you have an illness, gene therapy is usually used as a therapeutic method. So you can knock out a mutated gene or you can repair one. Or replace a mutated gene. But for this one, they're just introducing a whole new gene into the study. any questions. You can ask me personal questions.